Hey everybody, we'll give people a few more minutes to stream in, but before we get started, I just wanted to show you the puppy. This is Rosie. We got her last week and she's 9 weeks old and she's very crazy, but very cute. So hopefully you'll get to, as bonus incentive for coming to lab section, you'll get to see Rosie. So yeah. I'm sure this will mean that everybody comes next week, right? I'm just gonna give her to my husband really quickly, but be right back. All right, we're back. Welcome everybody to Lab 4. Hope you're having a great week. Honestly, because of with the puppy, I have gotten like only a few hours of sleep every night, so we're all just doing the best we can. Yeah, that's just life. Let me start sharing again. All right, can everybody see? I see the chat. Oh, yes, she's very cute. Can everybody see the PowerPoint slides? Okay, great. All right, so hopefully in the few minutes of music, you're able to pretend to be on a fabulous beach. Maybe some of you are on a fabulous beach, I don't know. In that case, I'm very jealous. But okay, so, before we move on to talking about this Lab 4, which was natural selection, super fun stuff, basis of modern biology, etc. Does anybody have any questions about pre-lab 4, post-lab 3, any of that stuff? Oh, nice. I see someone Miko's at the beach. Awesome. Okay, yeah. Any questions about, I guess let's start with post-lab 3 first. Any questions about that? I've gotten a few messages about if you weren't able to make it to lab session last week, what you can do. So you can definitely just do your own hypothesis on your own. I was realizing, I think it is really helpful to talk it through as a group and then to talk to me. So if you are able to, I encourage you, and you weren't able to go to lab session last week, I encourage you to come to office hours tomorrow morning, just to make sure that you're on the right track. If not, just listen to what I described in the last session video recording and you should be fine too. I have a chat. When are my office hours? So my office hours are always Tuesday and Thursday mornings from 9 to 10. And it's at the same link. All right, so not seeing any questions. I just wanted to go over the general question for lab 3. Okay, so yeah, so as a part of the post lab for lab 3, we talked about kind of narrowing into this, is hypothesis testing is the general subject. We talked about kind of narrowing into a hypothesis and then developing a methods for how to test that hypothesis. So there were different components to that. One of the kind of large scale thing that you want to think about, like the topic of your project, would be the general question. So in the example of the blepharisma as an organism, the general question was, how do different nutrient levels affect population size of the blepharisma? So that's the general question. And then a hypothesis would be that high nutrient blepharisma vials have a higher population after 14 days than lower ones. So that's, or yeah, sorry. Chat. Sorry, I'll look at that in a second. Does that make sense? I think, Crystal, you asked about the general question. So it's kind of the topic, the general thing that you're interested in looking at. So if you were looking at different foods and how they affect the population of tigers. You could be like, how do different nutrient sources affect tiger populations? And then your hypothesis would be more specific. Awesome. Okay. Yeah, okay, so those are the questions about lab 3. If you have any more statistics we're expected to do for post lab 3, there are no statistics for post lab 3. The statistics for post lab 4, there's like the average and standard deviation, and we'll go over that in a bit. But I don't think that, so for when you're doing the hypothesis testing, you're not actually going to do the experiment. And so Crystal, for the methods, that's like how you would do the experiment. And I really encourage you, if you haven't watched the recording for the lab section for last week and you're trying to work through the post lab, I really recommend going through that. That's going to explain like how to do the methods. And then again, if you can make it to my office hours tomorrow, that'll be really helpful. And also we can stay after and talk a little bit more about it also. Awesome. Okay, so kind of moving on to post lab 4, there were a few things that I kind of quickly wanted to go over that I noticed folks were struggling with a little bit. So for question 2 about natural variation, a lot of people said that natural variation was one of the requirements for variation was one of the requirements for natural selection, and that is true, but that was kind of framed in the question. They said, this is a requirement, so why do we need it? And the answer there was, you need a differentiation in success. And so if there's no variation, then there isn't like a differential in success or reproductive success. So then natural selection can't act on that population. So really a lot of people just said that it's a requirement, but we were really looking for why is it a requirement and what would happen if you didn't have it. For question 8 about the glucosinolates, the kind of stinky things in the plants, the question was also asking you to talk about how they would deter things from attacking the plant or eating the plant. So there were a lot of other general things about them that people said about, oh, they prevent disease in the plant, but really we're looking for like, how is it preventing predation? And so, yeah, these are just examples of like carefully, had a hard time finding things about the glucose. Yeah, basically they just make the plant kind of stinky and unappetizing. So that's kind of one of the main things. A lot of people talked about that it stopped insects from attacking them and other animals from eating them. Question 11.2, requirements for root growth. Let me make sure I get all of those. The main thing that people missed was space. So I believe there were 5. Let me double check. I just want to make sure that I say it right. So there were 5 of the things, and as long as you got 4 of them, I marked it as correct. But okay, yeah. So the things were that at the root level that are important: space, nitrogen, phosphorus, calcium, and water. So the things that weren't important are light and carbon dioxide. And that makes sense because the carbon dioxide is coming in from the atmosphere into the leaves, and then the roots can't get light because they're underground. So it seems pretty intuitive, but the one thing that maybe people weren't fully connecting is on the space. So root space is still important. For example, if you plant a plant and there's a ton of other roots under there and there just isn't as much space for them, then that plant is not going to be as successful. So yes, both root and shoot definitely need space. And a plant can choose to allocate like more energy into growing root or more energy into growing shoot, depending on the availability of nutrients in those areas, or space also. So yeah, but space is definitely important. Okay, so, and then For question 13, this was the last one where I saw mostly people were getting it, but just to clarify, they asked about the best possible conditions. So the best possible conditions for any plant would be no competition. So if a plant has unlimited space and resources, light, water, food, then they're going to be a very happy plant. So when they're talking about best possible conditions, it'd be mostly a plant is not dealing with either inter or intra-specific competition. So those were my main comments about this pre-lab. Okay, I will start sharing again. Oh wait, no, I'm still sharing, aren't I? Great. Okay, so we're gonna move on. I just wanted to clarify a few questions in the chat. You definitely don't, if you have to leave early, no worries, just watch. I would watch it on your own time later if you aren't able to make it to the whole lab section. And then a few people were asking me about if their plants are dead or they aren't growing or whatever, that's okay. They were hopeful that we'd get enough data from the students' plants that we can do our post-lab stuff on, but either way, you will have data to use. It's just fun to have your own plants and contribute to the class data set. So don't worry about it if the plants aren't working out. I know that, I mean, I study plants and I still have trouble getting plants to grow a lot. So I totally understand. So they don't need to flower. That's the thing, is that we don't really have time to do like a real reproduction study. So we're just looking at kind of proxies for fitness, looking at, you know, height, number of leaves, etc. So some of them are really short, some of them are taller, but it's just kind of, it is what it is. And they were all in different conditions. So this is by no means a controlled experiment. We're all just doing the best we can, as I said earlier, always. Okay, so kind of going broadly over the topic of this week's lab, we're going to be talking about natural selection. So you have gone over this, and this was talked a little bit about in the pre-lab, but talking about what the conditions are that are necessary for evolution by natural selection. So we talked about one of them in that question. Can anybody type in the chat or say out loud what those different 3 conditions are? Okay, yeah, so variance, exactly. That was what we talked about. There's variance, and this is a critical part. So you need to have phenotypic variance or genetic variance, et cetera, that has a difference in traits. Any other ones? What about those traits? So what is a condition for those traits in order for natural selection to act on the population? So they need to be phenotypically different, yes. And also, say you have this awesome, you're like really good at basketball, like amazing at basketball, and then you have a kid and you're good at basketball because you've trained a lot. Is your skill at basketball like the kind of variation that would lead to natural selection? Just because you've trained a lot, you're good at basketball? So it has to have a genetic component, right? It can't just be like a heredity, exactly. So it can't just be like Lamarck, he talked about, you might have learned about him in like high school, that he had this theory that the giraffes, they stretch their necks and then the babies have longer necks. Like he had this idea that things that happen in the mother's life will affect the genetics of the children, which mostly people laughed at him. There is some stuff about epigenetics and stuff, but for the most part, a trait has to be heritable for natural selection to act on it. So if you're talking about a famous experiment was looking at different mice coat colors, and different successes in different environments. And so the heredity of the coat color, the genes that the mother passes down to her babies, it's super important. Yeah, epigenetics is crazy and kind of throws a whole wrench into everything. But in general, so you have to have variation phenotypic variation, and then the phenotypes have to be inherited, like they have to have some genetic component to it. And then there's one last thing that is kind of intuitive and related to these two, but for example, like be able to reproduce. Okay, yeah, so be able to reproduce and at different rates depending on that trait, right? So the trait has to be related to the abilities, to the species' ability to be reproductively successful. Because otherwise, if it's just like hair color in people, for example, that's not a trait that's leading to natural selection. That's not a trait that like leads to differentials in success. So it's thinking about that. It's not just that there's variation, it's that the variation leads to different survival or reproductive rates, and that there's a genetic component. So those are the three. Does that make sense? I know that was kind of a long-winded explanation, but you'll keep getting introduced to these throughout if you're continuing to take biology classes, and if you take like EV 100 or whatever, you'll talk about this again. So these are really critical, those three components. So fitness, does anyone remember what fitness is? Not just like up in the gym working on your fitness, but like fitness in a biological context. So. Yeah, exactly. So fitness is how much you're able to reproduce, how many offspring you're able to successfully have. And then relative fitness is how many offspring you are able to successfully have in relation to other members of your same species or population. So for example, if we compare, we talked about elephants really quickly last week, that they take 2 years to have a baby, whereas like bullfrogs can have like 10,000 eggs, I don't know, at least once a year, maybe more than that. So to compare the fitness of those two creatures is crazy, because they're not like, it's totally a different level. So when you're talking about relative fitness, you're talking about relative to others of the same species. So yeah, fitness, not just getting swole, but like having babies. And then how might competition for resources affect reproductive success? Any ideas about this? You can throw it in the chat, but just maybe think about if it takes resources to successfully have an offspring. Think about when you're, pregnant and you need to eat for 2 or 10,000. If you're a bullfrog mom, like you probably need a lot of resources. And so if you're also competing against other members of your population, it might have negative consequences on your reproduction. And then kind of, we're also in this lab starting to introduce some statistical concepts, which I know statistics are confusing and frustrating, but they're incredibly powerful, and they're like really just the basis of everything. They're the basis of all these published papers, all the stuff that we know to be true really relies on statistics. So statistics are super important, and it's really awesome that you're getting this introduction to it. And how statistics can help us analyze our data is basically to tell us if it's significant or not, if what we're finding is a meaningful result. We talked a little bit about the COVID-19 vaccine last week and how important the statistics were in determining if the vaccine was effective. So statistics are important. And we're gonna start seeing if we can kind of detect differences in this little experiment that we're running as a class. Any questions about these concepts that we've talked about so far? Okay, awesome. So just as a reminder, general stuff, your homework. So you have the post lab 3 due tomorrow on Thursday, and then post lab 4 will be due next Thursday. And you're gonna turn in, as usual, a document with answering the questions, and then there's gonna be 3 graphs for the 3 different treatments. And then I think these are in the lab manual as well, but this is context and introduction of how to use a spreadsheet. Has anybody never used a spreadsheet before? Excel or Google Sheets, you can tell me privately if you want. But if you have never used a spreadsheet, I really recommend definitely check these videos out. And if you still need help after watching the videos, I really encourage you to come and talk to me in office hours, because it can be very intimidating, but but yes, exactly. Spreadsheets are very fun. They're extremely powerful. You can do so much math, so many formulas. It's great. You don't even need a calculator anymore. You can do everything in Excel. Or if you're me, you can do everything in R, because I learned Excel and I used it a lot in the working world. And then in grad school, everyone hates Excel and uses R. But I think they're all useful for their own things. So Excel is super important. All right, so context of how to use a spreadsheet, and then there's also how to do a t-test. So a t-test is kind of this basic statistical test to see if we can reject the null hypothesis, which is something we talked about last week, of being able to see if two groups in an experiment, two averages that we get, are actually different from each other. So this is a super important and really often used test for kind of determining if is there really a difference or is it just due to chance? Yes, when the p is low, reject the null. Loving the statistical lingo that we're throwing out here. Okay, so just a kind of review of the organism. I mean, hopefully some of you have been growing your radishes. If not, don't worry about it. But if you got the seeds, hopefully you've had fun kind of seeing the little dudes grow. We have the white morph and the purple morph. And so there's these 3 treatments that there was the instructions of how to do it in the lab manual, but treatment 1, no competition. You had 1 white and 1 purple morph growing on their own, not crowded, hopefully having all the resources that they could possibly need. Then you have treatment 2. So this is the intramorph. So all of the clumps are of the same morph, the same color morph. So there's 3 white together, 3 purple together, and then treatment 3, which is intermorph. So intra meaning within, inter meaning between. And in the intermorph, you had 1 purple and 2 white, and then 1 other box with one white and 2 purple, then 1 purple and 2 white. So kind of mixing it up. And if you have had success growing your radishes, just a tip for being able to identify which is which, especially in the intermorph competition one, the purple one should have kind of a purpley stem color. It's pretty subtle, but hopefully you'll be able to tell the difference, or even remember which ones you planted where. This is what we talked about, but just as to reiterate, and kind of I tried to get some illustrative pictures of it for the conditions for natural selection. We talked about phenotypic variation. So in the example of Hopi Hoekstra did all this experimentation on these mice, field mice. So you can see on the left picture, there's white and black mice in all across this area, but you can see the white mice look a little bit lighter on the sand, like it's easier to see the black mice on the sand, whereas on the rocks, the white mice really stand out. So phenotypic variation. And the trait must be heritable, at least in part. So I believe there's a few genes. They actually were able to isolate the genes in this experiment and figure out which gene it was that controlled the coat color of the mice. So it's a heritable trait. And then 3, the phenotypes, some phenotypes must reproduce more than other phenotypes. So differential reproductive success. And so in the case of the mice, for example, this bird is flying over and say it's like the dark colored rocks, he can see the white mice and he'll go and dive down and take them away. And so those white mice who are living in the dark colored rocks don't have an opportunity to reproduce and pass their genes on. So then there's more of the dark morph mice. So this is natural selection in action and kind of touches on all 3 of those critical parts for natural selection to act on a population. And then this differential reproductive success is what we are testing to a small extent in this experiment. Like we said, we don't have time to go through all of the reproductive cycle, but we're using these proxies for fitness. Okay, so the lab, question. Rachel mentioned with the dogs was an inheritable trait, but it was no phenotype. Sorry, I haven't watched this. So what was the example that Rachel mentioned with the dogs?
She mentioned that, oh, sorry, it's weird. I can hear myself talk. But basically, she mentioned this book where researchers went out and they tried to mate the friendliest dog of each generation until they got, or no, the wolf. They tried to mate the friendliest wolf of each generation until they got like a really friendly wolf to basically demonstrate how dogs were developed. But while I was watching the video, I was just kind of wondering how they statistically measured how a dog was friendly, because to some people, because to some people, like a friendly dog is completely different than another friendly dog. And when we think of behavior in humans, we usually do this test that has been statistically validated through several groups of people in terms of like, depression questionnaires, right? So I was just wondering, how do you like measure the friendliness of a dog?
Yeah, that's a really good question. I'm imagining that they had like these series of criteria, like how the dog reacted to like a stranger approaching it. Would they let you like handle it? Would they probably had like some checklist. That reminds me of, there's a really interesting radio lab that if you haven't listened to it, I recommend it, about this fox breeding program in Russia, that they tried to do the same thing to create these domesticated foxes.
It might have been that. I might have misspoke at some point. Yeah, it might have been that. Yeah.
Okay, cool. Yeah, that's a really interesting one. But basically, they like made these domesticated foxes in like a few generations by doing this breeding of the friendly ones. And also they like the foxes, foxes have pointy ears. Within a few generations, the ears became droopy. So yeah, it's like associated with being eternally a puppy and being friendly and docile. So super interesting. Super interesting. I mean, it's interesting when you bring in selective breeding, because it's not natural selection, right? Then it's like very choosy selection, but it has to be a series of traits that humans have arbitrarily decided are like better for the foxes or wolves or dogs or whatever.
Yeah, like eugenics in animals.
Yeah. People have done crazy things. I mean, have you ever seen how small like some chihuahuas are, like this big?
Yeah.
I mean, we've done crazy things with dogs, but all dogs are wonderful and we love them all. So anyway, yeah, thanks for bringing that up, Katie. So for the lab, we're going to be using 3 components of fitness, which are our proxies for fitness to estimate reproductive success, plant height. So plant height, average leaf length, and number of leaves are the proxies. You're going to watch a video to see how 3 components of fitness for each plant were measured, and then add your data to the Google form. So I think that's on the Canvas page. There's the Google form for you to, or maybe it might be in the lab manual also, the Google form for you to submit your data by. And please submit it if you can by Friday morning, because I think Pat is planning on sending out kind of a master spreadsheet of the data from everyone's plants by Friday afternoon. I think he'll try and send out another one on Monday, but just to give people more time to do this over the weekend, I would really encourage you to try and get it in by early on Friday. So yeah, you'll submit your measurements if your plants are able to succeed and grow on that Google form. Then you will be using Excel or another spreadsheet, Google Sheets. I think there are probably other ones, I just don't know them. To calculate the mean, the standard deviation, and the standard error for each component of fitness. You're going to use the t-test that we talked about to see if there is a difference in reproductive success among all 3 treatments for each component of fitness to answer the homework questions. So this talks about for homework question 2, was there statistical evidence that one morph had a higher relative fitness than the other morph? Use your results from the data table to help you answer this question. Then you'll give a detailed explanation that includes confidence limits, and we'll talk a little bit about that, but there's more information about what a confidence limit is in your lab manual. And then if you have conflicting results, you should describe them. So those 3 treatments, you're going to be comparing among the different measurements if you can see a significant difference among them in these proxies for fitness. Any questions about that? Okay. Great. So let's get statistical. This is kind of an example. I'm going to take us through a really brief showing of how spreadsheets work. So if you have Excel or Google Sheets, I kind of recommend you opening it just so you can get a little bit of a feel as I'm going through of how to use it. I'll do that in a second, but just kind of taking you through. So probably most likely what you'll get through me, but from Pat, is this lab section, our lab section with different data measurements. And these are, each row is just a sample. So pretend those are like your names of, you know, 1 is Katie, 2 is Crystal, 3 is Tina, etc. So your plants are these rows. And you'll have all the measurements that you've taken for your different plants. And it will be all of the measurements from all the different plants for our whole section. And you'll be getting an average. And the lab manual should tell you exactly how to measure, what to measure, etc. But for getting an average, you're basically going to do, and I mean, you've done averages before, you know, add them all up, divide by the number of samples, get the average. You might not have seen standard deviation before, but that is also super easy to do in a spreadsheet, and then getting the standard error for each of them. And I'll talk a little bit about entering this stuff, how to use formulas, and then what the standard deviation and error is. And then you're also going to be doing a t-test, and this looks terrifying, doesn't it? Right? Like, it looks very scary. It's not that crazy. So, but we fully acknowledge there are t-test calculators out there. So if you want to, you can use one of those. Otherwise, you can use the formula and just do it in Excel or wherever you want to do it. And then I'll kind of go through what these components are and hopefully make it less scary. This is another thing that we can work through in office hours also, but I promise you it's not that intimidating. There's just a lot of symbols in here. All right, so let's do the little spreadsheet demo just very briefly. Hopefully it's a recap for most of you, but so let's say these are for section A12. Actually, this is BO8, right? Whatever. My other section is A12. And these are the different measurements. So you'll have this list of numbers, and then just like the one on there, so maybe 8, 7, 5, 3, and you'll just go through. Hopefully they'll already be entered. If you want to do the average, you have to do equals average, and it will pop up for you. Average, and then you can just highlight. So I'm just holding down my mouse and press enter. Why is it a different number? Anyway, doesn't matter. This will give you the average from B2 to B11. So pretty straightforward. Just type in the formula, average, and the numbers you want to average. For the standard deviation, so if you've taken a statistics class, maybe this concept is familiar to you, but it's kind of the standard deviation from the mean of the samples, which also sounds jargony, but basically you're just seeing like kind of the spread of the samples that you're getting. And so this is another one where you can just type in the formula. So this is standard deviation to population standard deviation. There's 2 formulas in here. I think you could use either one. You're seeing me code very well. let's see. If they give us the same one. Slightly different. I would probably use the population one. Then the standard error is the standard deviation divided by the square root of the number of samples. So for this one, you'd have 1.73 divided by the square root of 10. Right. Yeah, so the square root of the number of samples. So whatever number of rows that we have with data in them, you're going to have your standard deviation divided by the square root of that number. So yeah, we'll be given the data. You just have to do the formulas and the calculations yourself. Okay, is this making sense? Okay. And again, just feel free to come to office hours and we can go more through the spreadsheet stuff. All right, and then, so kind of briefly going over the t-test. So the t-test, as I was mentioning earlier, is important for determining if there's a real or a statistically significant difference between two values. And so here, the null height, so let's say you're looking at treatment 1 and the plant height. So is 3.989 equal to or equivalent to, not statistically different from 3.889, which were the values, sorry. For these treatment 1, the green morph versus the yellow morph, this was from last quarter, so they were green and yellow-green instead of white and purple. The average for treatment 1, green morph was 3.989. The average for treatment 1, yellow-green morph was 3.889. And so our goal here is to see if those two are actually statistically different. And so the null hypothesis would say those are not statistically different. So this is not asking if they're different numbers. Like we can tell that they're different numbers. Is it significant? Is the difference significant? And so the null is that they're not statistically different. And the alternate hypothesis is that they they are statistically significantly different from each other. And so basically to get the answer of if we're able to reject the null, we'll take one from the other. So it doesn't matter which one, you're just looking at the absolute value. I mean, to make it easier, you can just take the bigger one from the smaller one. And then, so you have that as the numerator, and then in the denominator, you have the standard deviations of each squared divided by n. So you have standard deviation of group 1 squared divided by n plus standard deviation of group 2 squared divided by n, and then the square root of that addition. And then you divide the difference by that number, and it gives you this t value. And then, I know this is a lot of steps. With this t value, you use the degrees of freedom calculation, which is the n minus 1 plus n minus 1. So the two groups together, but n for group 1 minus 1 plus n for group 2 minus 1. And then there's like a t table here. And it gives you these different levels of confidence. And you're supposed to see if your t value is bigger than them. So our t value, 1.24, and we look at 16, which is that 9 minus 1 plus 9 minus 1 for the two groups, so that's 16. Look under 16, 1.746 is the 90% confidence that they're different, and 1.2 is smaller than that. So we are not able to reject the null in this case. The numbers do not appear to be statistically significantly different from each other. I know that was a lot, and I think that I would be surprised if anyone wasn't confused if this is the first time you're learning about that. So understand, hopefully by watching the video and reading, and then if you need to, coming to office hours, it can start to make more sense. But So basically you're doing this in steps: average, standard deviation, standard error. Then you're going to get your t-value. Then you're going to look on the t-table. You're going to compare your t-value to the t-table number, and you're going to see if the difference is statistically significant. Lots of steps, but this is part of the scientific process. So we're moving through. Last week we talked about hypothesis testing and making methods for your project. Now we're kind of on the other end of your project. Like, once you have all this data, how do you make sense of it? And this is a huge part of that. Are there any questions about this so far?
How do you do it? How do you find the formulas in Google Sheets? Like, I know when you found the average, you kind of highlighted everything.
Yeah.
And then you found the average. How do you do that in Google Sheets?
So if you just start typing equal and then average, does it come up?
So I've highlighted everything, and then
before you highlight, go to the square where you're going to do the average and press equal.
Is that like, what square is that?
So I can get 1 second. So we're almost done here, Katie. I can do that with you after the next one, and we can I can do a little demo for Google Sheets after I talk about error bars a little bit, and so we can go through that.
Sure.
Okay, good. Okay, so any other questions about what we've talked about so far? Awesome. Okay, so talking about graphs and error bars. So you're going to be turning in 3 graphs. You can scan graphs that you make. You can take a photo and insert into a Word document, or you can just copy and paste from Excel. That's, if you know how to make graphs in Excel, that's probably the easiest way to do it. But you can draw it by hand if you need to. And as with everything, and this is what I've been doing since undergrad also, is you can Google anything and kind of get like really, really helpful explanations of how to do things. Like, how do I make this kind of graph? I do it constantly all day long I do a lot of GIS and R stuff, and I'm always Googling, like, how do I make this table? How do I do this function, etc. So I really recommend that, but you can also always talk to me. So for the standard error, which we talked about was the standard deviation divided by the square root of the sample size, you're going to have a confidence interval, kind of standard error bar on either side of your mean, which is the length of one standard error. So basically, if you are looking at this graph, which is again from last quarter with the yellow and green, you're going to have the dots are the averages that we got, and then on either side you have +1 standard error and then -1 standard error. So if you've probably seen some scientific papers that have these kind of graphs, and the standard error is just kind of talking about uncertainty in the data. And so this is really important to see kind of the range of values. So if you can see, like on treatment 3, the standard morph had this really wide confidence interval. So this shows that there was a really big range in so this was maybe plant height. Yeah, a really big range in plant height for group 3 standard morph. So you're going to be plotting that. And basically the key thing is you're going to plot your points as the averages, and then have this +1 standard error line, -1 standard error line on the graph for each one. Okay, and then there's more detail about that also in the lab manual, and I believe that there's some in the video about it, but really there's a lot of good information in the lab manual about it too. Any questions about standard error bars and graphs? Okay, cool. That's it for what I had planned to talk about. If you want to stay on, I'll talk a little bit more about spreadsheet stuff, and then we can talk about anything else also. I can probably stay on for another 20 minutes or so. Good luck with this. This is a pretty challenging exercise, I'd say. Yeah, I've been studying statistics. I have taken a million statistics classes. I do them every day, and I still find them really confusing and challenging. So I 100% understand, like, this is just meant to kind of like dip your toe into the field of statistics. It's really cool and exciting also, but it definitely feels intimidating at first. So hopefully this helps clear it up a little bit. I'm going to, are there any questions in general about this before I stop the recording? And yeah. Okay, I don't see any in the chat. All right,